Continuing our discussion on the use of radiation technology in pest management, if you just recollect the list which I gave in the last class, majority of the insects belong to Dipterous insects and also Coleopterous insects, very few belong to Lepidopterous insect. One of the feasibility of using this SIT technology for Lepidoptera is very limited, because these Lepidopterous insects are highly radio resistant, because they need high doses for 100% sterility, mainly because of holokinetic chromosomes which are located on the DNA, which need high radio resistance, and more than that, because of the somatic damage which induces behavioral incompetence, that is the mating of the sterile insect is affected in the parent generation. That is why the success rate in Lepidopterous insect is very limited. To overcome this, a modified SIT technology or F1 sterility technique or inherited technique is followed in case of Lepidopterous insect pest suppression. Here the employment of F1 sterility is done very successfully, that is the use of F1 progeny which has got a substerile males in the parental generation itself is used using less amount of radiation. Even in Lepidopterous insect, males need a very low dosage compared to females for radiation for sterility induction. So this behavioral difference within the species can be used to our advantage. This induction of inherited sterility in F1 is very competitive with the natural wild males or the wild females. So inherited SIT technology is useful in Lepidopterous insect pest management. This can be better explained in this slide. Now in case of Diptera, we use fully sterile gamma dose, wherein in the parental generation itself the males will be sterilized. When they mate with the natural females, we find sperm altered with genome in case of a sterilized male, whereas the ovum is unaltered in case of natural female. The cross between these two leads to inviable zygote, which is 100% sterile in the parental generation itself. But this thing does not happen in case of a Lepidopterous insect, wherein we use a very low dosage for partially sterilizing using the gamma dosage. So inducing partial sterility in case of the parental generation. With this partially sterilized males, when it is mated with the normal females, we find the two cases. One is inviable zygotes are also seen with the dominant lethality gene. In the other case also we find viable zygotes which carry a recessive lethality. So when this is further mated with, this is considered as the F1 progeny. When this F1 male mates with the normal male, then we find again inviable zygotes in the F1. So that is why this has got the full sterility or the partial sterility in it. So this technique is most useful for Lepidopterous insects. Let us take an example of the successful case of the codling moth and try to understand how this SIT technology was most successful. Codling moth is an important pest of citrus, apple and pear in temperate regions extending from South Africa to Canada and parts of USA. You can see the amount of damage done to the citrus fruit. Almost 50 to 60% of the ripening fruits, almost ripened fruits are damaged, resulting in severe economic damage. Even the pheromone trap catches also indicate high number of moths per day basis. As I said, the 6 principles of SIT technique, they standardized the rearing technique using artificial medium, and then they rear it in these small glass chambers. Each chamber contains around 100 larvae, and they standardized the duration, that temperature, relative humidity, photo period required for multiplying the larval stage, and then they blow the adult which emerges from this rearing chamber. Huge number of adults involving both male and female are collected, and these adults will be later radiated using gamma cell 500 with a dose of 150 gray They have standardized this sterility dosage also, and then the radiated the adults are put into these small plastic containers with a hole, and they are stored in refrigerated situation of around 8 to 10 degrees centigrade before they are released in the ecosystem. They also add a lightweight sand particles, mainly to prevent the damage done to the adults, and they are further released into the apple trees using these small vehicles, and these adults are blown to the trees. Sometimes they also use small flights, helicopters for aerial distribution if the area is quite large. I have here in this table a successful figures indicating in one of the area wherein they released 2000 moths per hectare per week during the fruiting season with 7 to 8 releases, and they integrated this with the mating disruption technique of the pheromone. For 5000 hectares, the staff required is hardly 7 staff and 2 technicians. They were producing per day 60,000 adults per day sterile insects. See the figure here, number of sprays reduced from 12 to 2, very significant reduction in the number of sprays, and these 2 sprays were given only in the beginning of the season, mainly to reduce the population significantly. And the interesting feature is 100% of the share is from the Hortigo. This is a farmers owned group. They outsourced this technology to a company known as Entomon. They multiply the sterile insects, they undertake the release programs, and they charge almost 3000 rand, which roughly works out rupees 18,000 per year per hectare to the farmer. In terms of comparing the figures of the effect of this SIT technique, in Alfanseto River Valley between 2006 to 2013, if you see, with 10 to 12 applications of insecticide, they used to trap 13 moths per week per trap of the adults. After 5 years of this SIT technology imposition, it could come down to only 2 adults per trap per week. Such a significant decrease, which further came down to less than 0.5 adults per trap per week. Such a significant reduction in the trap catches we can see with imposing the SIT technology. Even in terms of economic damage to the final product, if you just see, it is less than 1% damage we see to the fruits at the packing level, in the packing storage conditions about the fruit sucking, and this with 10 to 12 applications of insecticide, even they have recorded 6.5% damage at the packing level. Such a significant effect we see because of this SIT technology. Even in pink bollworm in USA also, the results very clearly indicate the higher rate of success. If you see this table, the extent of refugia planted up to 2005 is around 37.4, which drastically reduced to 3%, giving room for more land for BT cultivation, which also increased the total cotton productivity. Then if you see the percentage of infestation of PBW, which used to be 15.3% up to 2005, which came down significantly, and the percentage of reduction is 99.9% reduction. If you see the pink bollworm moths caught per trap, it is around 26.7, which also gave about 99.9% reduction in moth catches. If you see the number of sprays undertaken for pink bollworm management was 2.7 before the SIT program introduction. After this introduction, total elimination of sprays was done. In terms of money value, the USA spends almost $18 million for managing the pink bollworm, which came down to only $0.17 million, which increased the net profit because of the SIT technology integration with the IPM schedule. This technology is in operation in India on various insect pests, particularly the SIT technology is undertaken by BARC on potato tuber moth, by Delhi University on tobacco caterpillar. And similarly, BARC again is working on red palm weevil. NRL is also working on another store grain pest, red floor beetle, and the F1 sterility, as I said, for Lepidopterus insect work is progress in bark as well as in Delhi University. UAS, University of Agricultural Sciences, Raichur has also undertaken the SIT technology for red gram Weber, which is an important pest of red gram. In addition to this, the radiation technology is also useful in disinfestation of stored products. So, radiation as a source of disinfestation measure for stored products which carry a lot of Coleopterous and Lepidopterous insect is one of the important technique. They have almost standardized 0.5 kg has been suggested as the safe dosage for the treatment for wide range of commodities, practically to control all these pests. Little higher dosage 0.6 to 0.7 kg is required to knock down the noxious mites from these stored products. Another most important thing is in the plant quarantine. The radiation technology has become a prerequisite for the plant quarantine. Anybody to export outside the quarantine program, the radiation technology is a must. It should carry a phytosanitary certificate wherein one should declare that the product that they are exporting has undergone the radiation treatment. Clearly, about 10 kg has been approved by the FAO and WHO as the safe for use against any food product. Similarly, 1 kg has been approved as a disinfestation for fresh fruits and vegetables. Unless they have this, the material will not be allowed to export. Certainly, the radiation technology is in the primitive stage in India, but has a lot of scope for its integration in IPM packages across the agri ecosystem, across the horticulture ecosystem, across the forest ecosystem in India. Thank you.